Planck Institute for Multidisciplinary Sciences, Germany. Dr. Singh has extensively studied the impact of herbs on human physiology, focusing on development, disease, and viral infections. Using single-cell biology, computational techniques, and epigenetic experiments, he explores the molecular journey of herb in health and diseases. He has pioneered methods for annotating, integrating, and analyzing the transcriptional regulation of the mobile at both bulk and single cell resolution.
Thank you. It will take a while to set this up. Yeah. So in the meantime, my talk is also on hypoxia. Very different from what you said. It's not about adaptation. It's intermittent hypoxia. What we get during doing any physical. Yeah. So I was saying. So I'm also going to talk about hypoxia more as molecular criteria to define the impact of hypoxia on our brains. It is not the adaptive ones, it is intermittent. And I think one thing I want to really mention that in the intermittent hypoxia, which you can give yourself for 3 hours in a day, let's say for 4 weeks, you have an enormous production of erythropoietin. So erythropoietin really increases your physical endurance. I don't know how much you remember about erythropoietin, but it was very, very popular in early 2000 when athletes who were going to Olympics, they were injecting it as a doping, and they were, of course, getting gold medals and all. So finally, when the console, I got. Yes. Okay. Okay. So, so I have a confession to make. This whole story was about human endogenous retroviruses. That is where I was showing the connection with lots of neurological disorders, including major depression, anxiety, obsession, compulsion. But just to maintain the integrity of the time, I'm not going to show you human endogenous retroviruses today, because as by the end of the presentation, you would know I have more interesting thing to show. So what I'm going to talk about is mental health of humans. This is the era of the crisis we are living when it comes to the mental health. Just before pandemic started, you see, as you can see, that mentally there is no country perfect in the world. Some countries are worse than others. Any country which is happiest, as you can say, if you will, at least have 10% of people suffering from anxiety, obsession, compulsion, or major depression. After pandemic, the WHO has released a pseudo data kind of thing, and that shows that it's the depression and anxiety rates are likely to be increased by 25 more percent, keeping the 10, 15, or 20% as a background, 25 more percent. It's a shame. It's not a good thing. We want our planet mentally healthy so that we can appreciate each other, we can be constructive, we can advance in the positive directions. Of course, we have to look into the brain. Very complicated, right? 100 trillions of the synapses, 100 billions of the neurons, And what is interesting that if you look at the evolution of the brain, while other organs, they consume 15 to 20% of oxygen in the regular basis, human brain, not only human brain, but all the tetrapods like mice and others, mammals, they consume only 6% of oxygen. What it means is that the brain, the neural networks, the synapse has evolved in the hypoxic condition. This is what we call hypoxia, when you have lesser percent of oxygen available to that. So what happens at the molecular level in the hypoxia? So as soon as there is unavailability of the oxygen, your PhD, this pro-hydroxylin domains, they are degraded. And once they are degraded, your hypoxia inducible factor, alpha, beta, 1, 2, 3, they are stabilized. So once HIF1 alpha and other hypoxia, HIF1 alpha is the master regulator, by the way, because once HIF1 alpha is stabilized, it targets other HIFs, HIF2 and HIF1 beta. So it is all, the promoters are targeted by HIF1 alpha. So it is very important to keep HIF1 alpha intact. So whenever oxygen is unavailable, the HIF1 alpha is induced, and it recognizes on your DNA this sequence which is called hypoxia recognition elements, HRE. And once it has recognized its sequence, it has bound, it triggers the transcription, and then you have the whole pathway or signaling cascade of the hypoxia switched on. Coming back to the depression, there was this very nice study with unknown mechanism, by the way. They had shown that intermittent hypoxia promotes hippocampal neurogenesis. That's a little redundant because neurogenesis is hippocampal anyways. There's no other region of the brain where neurogenesis happens. It's only hippocampal, indented gyrus. Produces antidepressant-like effects in the adult rats. So this group they had studied, they had given their rats 3 hours of intermittent hypoxia, and with the hypoxia, they have lowered it up to the 10% in this study. So they keep the oxygen level 10% for 3 hours, for 3 weeks straight, and they already see that anti-depression kind of phenotype in their brain. It's a very nice study. You see they have this MRI reports and others. It's very clinical group. So to push this frontier a little further, we wanted to test in humans, right? That is our ultimate goal. I took the single cell RNA sequencing data, the biggest single cell RNA sequencing data ever available for human depression. As you can see that they took this dorsolateral PFCs, this prefrontal cortex areas. That is where, so they had 71 subjects, 38 of them were females and 33 were males. And of course they were, you can fraction them equally, that how many of them were major depression disorder and how many of them were healthy. And they all went through the clinical parameters of measurements of depression. And I think more talks like Dr. Bansal also. So they showed that these are the cell types, any kind of cortex single cell RNA-seq data you analyze, these are the major cell types you always end up with. Excitatory neurons, inhibitory, oligo, OPCs, microglia, astrocytes, endothelial, pericytes, and mixed. So I'm not going to bombard here with lots of data, but I want to show you that there is one excitatory neuron, because this is... Oh, okay, sorry. So so among all the excitatory neurons, our focus is to really get the newly formed neurons, right? The younger neurons. The neurons which has capacity to mature, they would absorb all the pressure and stress and get mature, get migrate, get, make the synapse. So our target is that. So when we analyze this newly formed neurons from this hypoxia data, so they're not newly born, they're just newly formed because we can just track them according to their age. What amazed us, and it was unreported in that original publication when they produced the data, was HIF1 alpha, like at globally level, either it is male or it is female. The HIF1 alpha was the top down-regulated gene in the major depression patients. And HIF1 alpha, as you know, it is triggered by hypoxia. So here you see in the females, it was the top with the most false discovery rate and most down-regulation intensity. In males, there were some other genes, but HIF1 alpha is also the top candidate, which was down-regulated. So in our group, there were already data about the mice which were kept into the hypoxia chamber, and again given intermittent hypoxia. So we had followed the same protocol as this original paper was published. We kept our mice for 28 weeks in the hypoxia chamber with 12% oxygen. That was 3 hours per day for 28 days, for 4 weeks straight. And then we had single cell RNA sequencing from their hippocampus and cortex both. So just one result I want to show that how to read this trajectory. It is a pseudo-time trajectory. So every dot is a single cell, and this color means the time of their differentiation. The colors which are really green, they are very young neurons, and the colors which are dark blue, they have differentiated, so they are very old neurons. So in the same neuronal subtype, you can grade every single cell that what kind of age they have. And as you can see that these youngest form of the neurons, one is FOXP2 positive, one is QX2 positive, that is only present in the case when animals are under hypoxia. So the youngest group of neurons, they were induced by hypoxia, right? So the major finding from all this data, analyzing lots of things together, is that you have intermittent hypoxia that makes actually this was very interesting. So there was the number of mature neurons did not change at the significant level, but the newly formed neurons were populated to 200 more %. Or in the case of FOXP2 positive, it was exclusively present under hypoxia. And then you have immediate early genes when you have hypoxia, because HIF1 alpha also targets immediate early genes that are key for mood, memory, and cognition. But you have more oxidative radicals, then of course you have more radical vulnerabilities, right? Because they often cause DNA damage and cell cycle arrest and all kinds of things. So it's not a good to have lots of oxygen in the brain. We wanted to test if this intermittent hypoxia really can alleviate humans suffering from major depression disorder in the clinical context. So we had this hypoxia chamber for humans. It's a very nice chamber. You can have a Netflix there. You have a treadmill. You can sit. It is so once you enroll in the course, it is 3 hours every day. From Monday to Saturday, Sunday is off, and then for 4 weeks. And then, of course, we draw the blood from these patients before they go to the chamber and after they come out of the chamber, right? So just to measure everything before hypoxia and after hypoxia. Afterwards, these are the clinical kits, right? With the BDNF, serotonin, microRNAs and all, where we can really test how their level of depression, anxiety, or obsession compulsion after they come out of hypoxia. So far there are 17 volunteers, and we can clearly see that anxiety, obsession, compulsion, and major depression is significantly improved after they have gone through the hypoxia chamber. So this hypoxia chamber, and this is what making me think, is that this only reproduces athletes' high altitude training. So athletes, when they go on high altitude training, they also reprogram their system in this certain way, what we are trying to deliver them into the hypoxia chamber, right? So also it is anti-aging. If it is giving you lots of new neurons, replacing the old neurons, it's very anti-aging. It just rejuvenates your neuronal network. With a high altitude, again coming back to erythropoietin. So this is what you get when you go on the high altitude training, when you rehearse for your Olympics and all, you get more erythropoietin naturally. So the athletes, what they used to take as a doping in early 2000, now you are giving them by offering high altitude training, right? So erythropoietin is very important, and this is what distinguishes between intermittent hypoxia and chronic hypoxia. So the chronic hypoxia is what you were talking about, ma'am, that you get adapted on high altitude. This is the different measurement. So here you expose the individual to the hypoxia, you let brain feel the challenge, and to respond that challenge, they produce new neurons. And as soon as these new neurons are ready for the combat, you take this challenge back. And once you have taken this challenge back, these new neurons, they are targetless. They do not have goal. So what our hypothesis is that these new neurons would go to the synapse and replace the bad neurons or the old neurons, which are having the toxicity and all. So what we did, of course, we cannot integrate HIF1 alpha into the mouse genome and do the experiment. So what we have to do, we had to rely on the erythropoietin. So erythropoietin is induced upon intermittent hypoxia without triggering VEGF-A and other oncogenes kind of thing. So this was very safe. So we did the experiment with the erythropoietin. As you can see that after hypoxia, HIF1 alpha is induced, and it targets the erythropoietin. So erythropoietin expression is induced 6 hours after you give the hypoxia. So one of the first targets, right? So again, we did our experiment in the same way. So we took the 28 weeks old mice, we injected erythropoietin on every second day, and after day 49, we took their hippocampus, and we did multi-omics. We did single cell RNA-seq and single cell ATAC-seq both. So I will show you just one data for single cell RNA-seq, one data for single cell ATAC-seq. So what happens after erythropoietin? This one particular kind of neurons, which are lost upon in depression patients and others, they are populated 200% more. So these are newly formed, migrating, superficial. These are newly formed, migrating, firing, serotonergic, like producing serotonin. These are newly formed, migrating, superficial, and serotonergic. So the neurons which produce serotonin, they are enriched 200% more when you don't treat them with erythropoietin. And of course, all you need is serotonin to elevate from depression, right? So this was, and then from the ATAC-seq, so one of the major questions everyone asks, what is the mechanism? I mean, adult neurogenesis is very rare, and you have to give very strong stimulation, like in vitro kind of condition, to trigger the adult neurogenesis. How does this 200% neurons enriched in the hippocampus, in the dentate gyrus area? So when we did the ATAC-seq, single cell ATAC-seq in this particular lineage, the newly formed, we found ASCL1 gene. ASCL1 gene, the chromatin around transcription start site, was only opened under erythropoietin. And you know what ASCL1 gene is? It is for the transdifferentiation. Actually, you just take the two genes, one ASCL1, one NGN2, put it into the astrocytes, and astrocytes become neurons without going to the trajectory of stem cells. So this is the direct conversion. And when we look for the transcription start site of the genes and the transcription factor which are enriched, only after the erythropoietin induction, there also we see ASCL1, lots of immediate early genes which I discussed before, right? So this is what erythropoietin does, right? So this is the canonical way of differentiation. When you have neuroprogenitor cells or embryonic stem cells, they become neurons with lots of neurotrophic factors or transcription factor. When you give erythropoietin or hypoxia, because erythropoietin is just not possible without hypoxia, right? So when you give hypoxia, these cells transdifferentiate. These astrocytes, they start becoming neurons. And we think this is the reason why you have 200% new neurons. And it has a huge implications in the regenerative medicine, not only in regenerative medicine, but also in neurobiology. Because in vivo transdifferentiation is very rare. So they have shown one paper from Martison et al. If you see, they say that, oh, if you have a stroke or if you have a brain injury, then only astrocytes are stimulated, and these astrocytes are stimulated to make the new neurons. But here we are stimulating astrocytes by hypoxia and erythropoietin, making new neurons. So this is our hypothesis, which we tested and which I have shown, that this is the neurogenic hypothesis of depression. So in depression, your old neurons, they're toxic. How to replace them? Give them the new neurons. If you have new neurons, they would replace the old neurons, and your synapse is rejuvenated, your synapse is faster, your synapse is fitter, your brain is renovated in its performance, and you feel like, you feel amazing. I had been to hypoxia chamber, by the way. So with this, I would like to thank everyone who was directly or indirectly associated with this project. Umut, my PhD student, this is all work from this one, this computational biology, right? Our supervisors, Klaus Nave and Hannelore Ehrenreich. We also have a collaboration. We are doing the transdifferentiation assay. We are differentiating astrocytes to the neurons from Cornell University, where my two ex-PIs, Doug and Cedric, are there. Lara, we are collaborated on multiple sclerosis and Alzheimer's disease because we think that this could also be the cure for these neurodegenerative disorders, right? So with this, thank you so very much.
Thank you very much, Dr. Manvendra Singh. It was really a brainy one. So I enjoyed because this kind of depressions and whatever you were talking about nowadays is coming up in the world, and we can take one or two questions going up. Yes.
Whenever you say that whatever you were talking about, I know how much you enjoyed.
Thank you.
In your cases where you had a reduced expression of one alpha, did you also see any changes in expression of EGLN1 or EGLN family of genes?
I'm sure I have come across that gene, but I don't remember back of my head.
Please.
The benefit is sustainable. Sustainability of this effort after you.
Great question. Yeah.
How long?
Great question. So so it has been 9 months to 6 months till these patients came out of the hypoxia chamber. The bar plots where you see it's a modest, very modest difference. After 6 months, 3 or 4 of the patients, we had a chance to get their blood again. Now the differences are remarkable, dramatic. So it seems like at once it just triggers the neurogenesis, then it does its job. It's like a wheel, you know. You push the wheel, it just goes down the thing. So I wish I could show you. We are not allowed to show that data anymore. I wish I could show you. That looks remarkable. What happens after 6 months of hypoxia treatment? Yeah.
Dr. Manvendra, I have a question for you. Because I have treated a lot of patients with hypoxia, neonatal hypoxia. So if by your things, if I put it into that, then I find that those hypoxic babies should have more brain matter.
No, ma'am, because it is only for adults. Because in kids, brain is neuroplastic, right? The brain is already generating new neurons. So it is for the condition when brain fails to generate new neurons, then you give some extra cellular stimulation. The kid's brain is very neuroplastic, so I don't think this. We don't know, of course, but if you ask my person, I don't think.
How do you differentiate between new neurons and old in histology?
Oh, in the histology, so the old neurons would have more spines, more interactions. Their branching would be so huge and long and thin. The new neurons would have little less branching. It would be like a little plant on the pot. And the big old neurons, of course, are like the trees. So when you look at their histology.
Thank you so much.
Ma'am. Mini ma'am was my first mentor. Yes.
This is really a good question for the brains you are coming up. So these are brains.
Fantastic, you know, we have attained too much worry when we started talking about bioinformatics, but now we know who does the Thank you.
Always so charming.
Neurogenesis must have taken.
Of course. Yeah. I mean, any physical active activity that makes you breathless, right? So it could be running really fast, it could be biking really fast, it could be, lots of other things. Where you feel breathlessness, you just have to challenge your brain that there is some threat of the loss of oxygen. As soon as they produce new neurons, you just have to take the challenge back, and then you feel awesome afterwards. I mean, if you dance, you feel great after dance, right?
See, the understanding here was.
I know what he was asking. I know.
I feel this after this everybody got a new bunch of neurons because so it was really wonderful session I have. So I have just a kind of small interaction with you that you are talking about the hippocampus and the prefrontal. So what about amygdala? This is a kind of fear one in the brain. So does it has any kind of connections with this hypoxia?
No, hippocampus and especially the CA region where you have dentate gyrus, that is the point of interest because that's where neurogenesis happens. So rest of the areas, if neurons migrate from there to cortex or to amygdala, that's a different question. But if we want to see the effect, if we want to see the difference, that how much is really happening, that is the core area, like hippocampus, dentate gyrus area. Yeah.
So this was a really very illuminating session. I have learned a lot. So I think we can end the session over here with a happy note. Thank you so much. So if you can just make a summarization, I think Madam can give a summarization of the whole aspects.
It was really a very nice session, which started with, and just for Manvendra, I would like to can you correlate with these neurogenesis to plasticity of the brain? Because we all know
Yes.
that adult brains are not plastic in nature.
Yes. Oh, yes. So as I said, the trajectory is different, but the end product is the same. So there is no neural stem cells in the adult brain, but it has enormous capacity of making astrocytes into neurons. So at the end of the day, you want new neurons. There is a different way how they come, but you have it in the end of the day. Thank you.
So now I'm going to summarize today's session. So we started with the presentation of Dr. Srikanth Mane. So he enlightened us on the topic increasing diagnostic rare
We
in
are
rare
dying to do that.
diseases,
We are dying to do
lessons
that. They
learned
won't give us
and
permission so
defining
easily.
variants of unknown significance. So
Yes,
in
it
his
is.
talk,
It is.
he
It is. You
explained
just give
about
me a
all the facilities
But
and initiatives
get me
which were
permission in India. I would like to do that.
available for genomic analysis. And he also told us and briefed us about that there are various ways by which muscular dystrophy
अरे जा कहाँ गए?
diseases
हमारा
can be
felicitation हो गया.
delineated, and for that functional characterization of various variants using exome genome sequencing. So first he started with the exome genome sequencing, and then he mentioned that other than there are various challenges and limitations of these sequencing techniques, and finally he came to the some of the he introduced about the new genome optical genome sequencing like optical genome mapping, uh, long read genome sequencing, and then he also mentioned about the new concept where he introduced us about the RNA sequencing, which has related with the cases diagnosed using RNA sequencing. Though there are lot of limitations with various tools and technology in genome diagnosis, but there should be a comprehension between the genome function and precision medicine making. So now we totally go for the wider genome sequencing technologies, which can be helpful for identification of various rare diseases, including new modern technology. Second talk was delivered by Dr. Pankaj Khurana from DRDO, and it was just very good idea for the youngsters and young scientists that they came to know about that DRDO is also the center where 9 life science departments are working, and so I think now our young investigator will come up to apply for various project position and funding, and faculty members will be encouraged to apply for funding with to the DRDO, as Madam has shown us the way to go ahead. She, in her talk, she introduced about the various computational approaches for high altitude health and performance optimization. So which was very important for our soldiers which are residing on high altitude. And there she has talked about that 7 specific mRNA, miRNA were introduced by these data integration and whole human genome. And these 7 miRNA can act as a future miRNA medicine. So we got another opportunity to have the regenerative medicine or new medicine technology to come up in future. So now we can use miRNA as a, instead of tablets, we can go for miRNA technology as a medicine. So now
The third talk was delivered by Dr. Manvendra Singh, who enlightened us about the hypoxia and its role in the neurogenesis. So already we know that. So through his talk, we all came up to know that now we cannot make adult brain as it is still can be made as a sorry. Adult brain now can be made plastic by having hypoxia treatment. So to rejuvenate, to have a more neurogenesis, we have to do intermittent exercise